All right, everyone. As I'm waiting for the slides to pop up, there we go. So last class we focused, we wrapped up the protein trafficking lecture and tackled glycolysis. One thing that's key for us to note is that I want to take a moment to spatially situate ourselves with where we've been and where we're going. If you guys remember from our lecture talking about diabetes, when we ingest food that has glucose in it, glucose is absorbed, and I imagine some of this is going to dovetail off of some of the content you all have learned in Physiology 1 if you have taken or are taking that class. Typically, glucose is absorbed in the small intestines and absorbed into the bloodstream. And then using our info some of the info that we talked about with the diabetes unit, or not unit, the diabetes lecture, there's going to be a response to blood glucose levels going up where insulin is released that enables glucose to now be shuttled into our cells via glucose transporters. Glycolysis is happening within the cytoplasm of the cell. That's where we've been so far. A lot of the enzymes that are important, excuse me, all of the enzymes that have been really critical for metabolizing glucose into pyruvate, two pyruvate molecules, is all happening within the cytoplasm. Today, we're going to be focusing on two other key steps, two other key processes that are going to be important for oxidative phosphorylation, the entire aerobic respiration that happens after glycolysis. And all of that's going to be taking place within the mitochondria. There's going to be a few different key compartments of the mitochondria that we'll be focusing on today. The inner compartment of the mitochondria is called the matrix. You can think of another way to think of that as like the cytoplasm of the mitochondria. The inner part is called the matrix. What's also unique about the mitochondria is that it's a double membrane organelle, meaning we have an outer membrane and an inner membrane. What's going to be key is where there's going to be a lot of key molecules that we'll be talking about that are a part of the inner membrane that actually shuttles ions into the space in between these two membranes called the intermembrane space. So we'll be moving from the cytoplasm into the mitochondria today and working with with our intermembrane and the intermembrane space between our mitochondria. What's key to note is that the mitochondria are key to aerobic respiration, and that this is only going to be occurring in eukaryotes because only eukaryotes possess mitochondria. The two major steps that we're going to be focusing on for aerobic respiration today might be things that you've heard of before, and we'll be able to kind of piece the story, we'll situate these processes within the scope of metabolism. The first is going to be the TCA cycle. It goes by a few different names. So TCA stands for tricarboxylic acid. There's going to be 3 carbon dioxides that are released either in preparation or during this cycle. Which is why it bears its name, the tricarboxylic acid cycle. It also goes by the Krebs cycle or the citric acid cycle. What's going to be key is that the TCA cycle is going to serve a couple different key functions for us. One, it's going to generate more NADHs, more reducing equivalents, that these coenzymes that serve an important role of being acceptors of electrons that carry them over to their final destination. What's also really important for the TCA cycle is that there's going to be opportunities for entry points into metabolism for amino acids. So typically, you can break down the food that we ingest into 3 basic molecules. We have glucose, protein/amino acids, and lipids from fats. All of these are going to be enabled to be plugged into metabolism or these metabolic processes at different points. So for amino acids, they'll be enabled to be plugged into the metabolic pathways through the TCA cycle. At least that's one entry point. The last process that we'll talk about is oxidative phosphorylation. Another name for this is the electron transport chain. The 2 key functions that oxidative phosphorylation, or OXPHOS as it's sometimes dubbed, is that it's going to help create a proton gradient via oxidation of some key coenzymes. And this proton gradient is going to be really key because it's going to help drive the synthesis of ATP. There's going to be an actual ATP pump or ATP synthase that's going to be enabled by by shuttling protons across the inner membrane here. So again, as we talked about the TCA cycle, what's key to note is that it's where we take pyruvate that's been generated from glycolysis, and it's going to be shuttled into the mitochondria. And there's going to be kind of a preparatory step that's pretty key that helps prepare us for entering into the TCA cycle. The first step is going to be we have pyruvate up here at the top. That's going to be oxidized via pyruvate dehydrogenase right here. It's going to generate our first carbon dioxide. It's going to generate our first reducing equivalent right here. And it's going to generate a molecule called acetyl-CoA. Acetyl-CoA is the key molecule that helps jump us into the TCA cycle. What's important to note is that there's going to be a lot of steps, a lot of processes that are involved in the TCA cycle. Similarly with glycolysis, I'm not asking you to memorize the entire TCA cycle. I'm not going to ask you to memorize each enzyme. What I am going to ask you to take note of when you study for the MCATs, you'll get a chance to memorize all of this, which Ahav will be taking very soon. So what's important to note is going to be the inputs and outputs of the TCA cycle. So we have pyruvate that's been oxidized via pyruvate dehydrogenase, generating acetyl-CoA. And acetyl-CoA is going to jump into the TCA cycle here. From there, there's going to be a few key steps that are important to note. So we have what's also helped us identifying how many carbons sometimes are in the metabolite, depending on what step you're in during the TCA cycle. So, for example, with pyruvate, we go from a 3-carbon chain to a 2-carbon chain. Here, where we have an acetyl group and coenzyme A, or CoA here, and then via the first enzyme here, we'll use oxaloacetate and acetyl-CoA to generate citrate here, where we have the generation of our first water molecule here that's released. CoA is going to be released with this thiol group there, and again, citrate is going to be metabolized into isocitrate. And then here at this next step, we have our next oxidation step where we generate another NADH reducing equivalent, our second carbon dioxide that produces alpha-ketoglutarate. From there, we have another oxidation event that produces another carbon dioxide. And so what's important to note here is that each carbon dioxide that's generated is generated via an oxidation step. But what's also important to know is that not every oxidation step is going to generate a carbon dioxide. They're not, it's not mutually exclusive in that way. And so after metabolizing isocitrate into alpha-ketoglutarate, we have this oxidation step where we'll get succinyl-CoA. What's also important to know is that we're also going to have, via this metabolic step, there's going to be the generation of an ATP. We'll talk about this a little bit more in a second, but there's going to be the phosphate there can be used to generate an ATP. From there, we have the generation of succinate. That's also going to lead to the generation of a different species of a reducing equivalent. Instead of NADH, we're going to get an FADH2. It's not exactly the same, but it's another reducing equivalent. And we'll actually talk about later in this lecture what are the differences between NADH and FADH2 when it comes to feeding into oxidative phosphorylation. And so that step will produce fumarate. From there, we'll undergo another metabolic step where we'll get malate, and from there we'll go back to oxaloacetate, which again, if you remember this feeds. This was one molecule that can feed into gluconeogenesis, but this brings us all the way back to the cycle where we're waiting for another acetyl-CoA to jumpstart the cycle again and to go through the entire process. This process is going to be consuming water. It's going to be yielding carbon dioxide and yielding key reducing equivalents that are going to be important inputs for oxidative phosphorylation. coenzyme A is going to be critical for is going to be a critical molecule where it serves as a shuttle for the acetyl groups to form acetyl-CoA that plugs into the TCA cycle. Somewhat similar to NADH, its structure bears an adenine with a ribose group. We have a pyrophosphate bridge. We have pantothenic acid here. In case this might sound familiar to some of you, that's actually vitamin B5. So there's going to be a few key molecules are actually vitamins that actually help promote and facilitate metabolism. So the key take home there is going to be take your multivitamins. From there, we have our sulfohydryl group, or the thiol here, that's going to mediate the addition or joining with the acetyl group here. And so this is all CoA that's going to help facilitate the entry into this TCA cycle. What's also going to be critical is that coenzyme A is going to be really important for fat oxidation. Again, we're going to be talking about how not just glucose, but amino acids and lipids can get plugged into metabolism to generate ATP and generate energy. So this is going to be a key substrate for fat synthesis in some cases, but also it's going to be a key substrate in enabling the production of fat oxidation. So the entry point, I guess I'll actually take a moment to stop there and give space for a couple of questions. Are there any questions yet? All right, we can go ahead and jump in. So again, the entry point for the TCA cycle requires pyruvate being oxidized into acetyl-CoA. This is going to yield our first NADH. It's going to yield, again, the first carbon dioxide for the tricarboxylic acid cycle, the TCA cycle. What's going to be key to note is that the acetyl-CoA, we go from the 3-carbon chain to a 2-carbon chain to synthesize acetyl-CoA. One of those carbons from pyruvate is donated to produce the first carbon dioxide. So the 2 carbons are going to be donated from acetyl-CoA. The 2 carbons are going to be donated to oxaloacetate to help produce, in addition with the hydrolysis of water, to produce citrate. For the subsequent steps, we're going to have our first oxidation step. That's going to yield an NADH. That's going to yield a carbon dioxide to produce alpha-ketoglutarate. Another oxidation step that's going to go from alpha-ketoglutarate to succinyl-CoA. And then the subsequent step, again, will produce a GTP. The production of that GTP can lead to the production of ATP. Via these dinucleotide transporters can help mediate the donating of a phosphate group from GTP to ATP. And so, but it's a dynamic process by which it may or may not, it's a dynamic process that's in flux, whereby sometimes it might be a GTP and sometimes it could be an ATP. But again, we have, at this point, we now have our 3 carbon dioxide molecules that have been generated. And again, we have, going to fumarate, we have the formation of a new species of reducing equivalent called FADH2. It is also an electron acceptor. We'll talk about later how it's different from NADH, and it's going to be a key difference for us to note based on how efficient it is at facilitating oxidative phosphorylation. And finally, with this final oxidation step, where this NADH undergoes reduction to become its reducing equivalent, we get back to oxaloacetate. That's going to put us, that's going to get us ready to accept a new acetyl group from acetyl-CoA and jumpstart this entire cycle again. For the second species of the reducing equivalent, FADH or FADH2, it's going to be flavin adenine dinucleotide. So again, similar as before, we have our adenine group here. We have our ribose sugar pyrophosphate bridge. What's different is that this is going to be riboflavin, which is vitamin B2. And so when it undergoes reduction to become its reducing equivalent of FADH2, we have the addition of the two hydrogen ions here and here in its reduced form. So to summarize what the take home is, what we get from the TCA cycle. So we're putting in acetyl-CoA, and from acetyl-CoA, we're going to generate, and this, what I should say is that this is excluding the preparatory step of pyruvate becoming acetyl-CoA. So this is just jumping into the cycle. So from one acetyl-CoA molecule, we get 2 carbon dioxides, 3 NADHs, and 1 FADH2. It's important to know that for every glucose molecule, we're getting 2 pyruvates, which will become 2 acetyl-CoAs. So for total inventory of what's happening here, and if we include the preparatory step, the intermediate step of pyruvate becoming acetyl-CoA, we're going to end up getting overall 2 ATP. We're going to end up getting 6 NADHs. But if we include the preparatory step, that's going to become 8 NADHs, because we get 1 NADH every time pyruvate becomes acetyl-CoA. We get 2 pyruvates from glucose. So that overall reducing equivalent number is going to be 8 NADHs and 2 FADH2s, as well as 2 ATPs. Now, if we combine this with everything that's happened in glycolysis, so far, our net yield of 1 glucose molecule, when we've done glucose, the intermediate step of pyruvate becoming acetyl-CoA, and the entire TCA cycle are going to give us 4 ATP, 10 NADHs, and 2 FADH2. So what's going to be key to know is that we're going to take NADH and FADH2, and those are going to be used in oxidative phosphorylation to generate a lot more ATP. I'm going to stop there and give space for a couple of questions now that we've wrapped up the TCA cycle. Yes. Yeah, so for that slide, so what's going to end up happening is that we have our 2 carboxyl groups here. Sorry, here we go. We have our 2 carboxyl groups here. The water is going to be used where we lose coenzyme A, and instead we're going to generate, we're using water to generate the different hydrogen ions that situate around and help form citrate. So it's used in the water molecule is used in this metabolic step to generate citrate from oxaloacetate and acetyl-CoA together. No, it's maybe a helpful way to put this is, let me see. So the combination of acetyl-CoA coming in, oxaloacetate, and this water molecule all together lead to the formation of citrate and the loss of CoA with this thiol. So the CoA was bound to this acetyl group here via this sulfur here. What's going to end up happening is that coenzyme A is going to be separating out one of the hydrogens from water, is going to create this sulfohydryl or this thiol group here. So that can leave away and stably leave. And what's going to end up happening is that now that one of the hydrogens are donated, it's going to be a hydroxyl group now. That'll help form with oxaloacetate and now just acetyl, this citrate molecule. Does that help clarify? If it doesn't make sense, that's okay. So if that was a simpler way to think about it, is just that you're separating the water molecule and using the constitutive pieces to help form with acetyl-CoA and oxaloacetate to create citrate. So if you cover up, so we have CoA with this thiol, the sulfohydryl. So if you cover this up right here, this is an acetyl group right there. So you have your acetyl group and oxaloacetate. They're going to come together, and you're going to use one hydrogen is given over to create this thiol here, the sulfohydryl. What's left over is the hydroxyl group. You have OH. So now you have OH with oxaloacetate and this acetyl will form citrate. That's what ends up happening. Does that clarify? Yes.
On the most recent slide, there's like a net yield for glucose so far. Is that just like glycolysis plus TCA?
Thank you. Glycolysis plus the in-between step of pyruvate becoming acetyl-CoA and the entire TCA cycle. That's what the net yield per glucose is so far. That's what that means. Yes. Yes, because one pyruvate to one acetyl-CoA yields one ADH, but we get two pyruvates from glucose. These are great questions. Yeah. Yes, it is. So both NADH and FADH2 are both coenzymes. Their main distinction will show up when we get to oxidative phosphorylation, but they are both coenzymes that both serve in their reduced form as an electron carrier or electron acceptor to then be to later donate them in the next process we'll talk about. All right. So to this point, the TCA cycle has been happening within the matrix of the mitochondria. Again, you can think of the matrix as almost like the cytoplasm of the mitochondria. We're now going to be entering into the electron transport system that's happening at the inner membrane of the mitochondria. This is where the electrons are finally going to leave our coenzymes and help mediate their key function here. So far, NADH and FADH2 have been accepting electrons. They've been undergoing reduction and entering into the reduced equivalent form. Here is where they're now going to pass and donate their electrons off, and now they're going to undergo oxidation themselves. They've been the recipient of electrons at every single oxidation step, but now they're the ones undergoing oxidation, and now they're the ones who are going to be losing their electrons. What's going to end up happening is that we have a few key, what we call complex proteins in the inner membrane of our mitochondria. We have complex 1, complex 3, and complex 4. We also have here a key protein, macromolecular protein that's called ATP synthase or the ATP pump. This is going to be a key protein that helps, a key structure that helps with the generation of ATP. So what's going to end up happening, we're going to have NADH undergo oxidation here, and it's going to donate and lose its electrons into complex 1. We're going to have these carriers that are going to pass along these electrons from FMN to iron sulfate over to coenzymes that then pass them over where you see CYT, that means cytochrome, these cytochrome proteins, and they'll continue to pass them over. What's going to end up happening also is that whenever these electrons move into the complex or are passed over, we're going to end up having the movement of hydrogen ions across the inner membrane into that intermembrane space. This is going to be key. Again, when we talked about what's important, what are the important roles for each process, the movement or flow of these hydrogen ions into the intermembrane space is going to be really critical, because the ATP synthase is going to generate ATP for every 3 hydrogen ions that it uses and moves across. So with complex 1, we have 4 hydrogen ions that move across there when NADH is oxidized and donates its electrons. As the electrons move from coenzyme Q over to cytochrome B at complex 3, we get 2 more hydrogen ions here and 2 hydrogen ions when coenzyme Q interacts with the electron. So we have 2 from coenzyme Q and 2 from complex 3. So overall, so far, taking inventory, we had 4 hydrogen ions move across here, and altogether 4 more hydrogen ions move here. As these electrons continue to be passed over, we get to cytochrome C here that moves it to complex 4. We have 2 more hydrogen ions move in. So we get 10 for every NADH here, we get 10 hydrogen ions that have moved across into the intermembrane space. And finally, with a hydrogen ion here and the presence of oxygen, we're going to use that electron to generate a water molecule here. Again, the key thing to take home here is that NADH is now undergoing oxidation itself. We have the movement of the donation of the electrons to these complex proteins in the inner membrane of the mitochondria. The donation of those electrons are now going to start driving hydrogen ions across these complexes, or as a result of coenzyme Q here, passing along the electrons, they're going to move 10 hydrogen ions across for every NADH molecule. About 11 years ago now, we finally got the structure of some of these complex proteins and what they actually look like. What ends up happening is that we have these, what was determined is that there's actually these electron pores present within these complex proteins at the inner membrane of the mitochondria. So what ends up happening is that when, excuse me, when the complex proteins are in its oxidized form, there's a channel that opens and closes. In its oxidized form, it's opened towards the matrix, but when it's in its reduced form and electrons have been received, the complex proteins will then open up their electron pores to enable the flow of electrons and enable the flow of hydrogen ions here. And so it's the couple of key first molecules that help mediate this are flavin mononucleotide, that's FMN. And we have, again, we have complex 1 that's going to open up and enable the flow of hydrogen ions that enables these pores that open up that enable the flow of the hydrogen ions that then is going to subsequently open up as each oxidation event occurs, as electrons are passed over and received by subsequent coenzyme Q or subsequent complex proteins at the inner membrane. So all of this is going to end with the electrons being used to generate a water molecule and find its final destination at an oxygen molecule here. And so one other key thing is that we talked about complex 1, 3, and 4. You're probably wondering what and where is complex 2. Complex 2 is the membrane protein that FADH2 is going to interact with. So a key thing to note is complex 1 interacts with and receives electrons from NADH. Complex 2 interacts with FADH2. But one key distinction is that FADH2 is not going to generate complex 2 to move 4 hydrogen ions across. So that means that FADH2, while it's going to undergo oxidation and donate electrons here to the electron transport chain, it is a less efficient coenzyme as it generates 4 less protons to move across into the inner membrane space. And those hydrogen ions, again, are important because the ATP synthase or ATP pump uses 3 hydrogen ions to turn its gear and generate an ATP molecule. So if FADH2 if it's moving less hydrogen ions across, it's a less efficient way to generate ATP. So the ATP synthase, some of the building blocks for it is a combination of this F1 protein complex, F0 protein complex, and this stalk right here. When they're assembled all together, they are what makes up the ATP synthase. You can also call it the ATP pump. What ends up happening is that every time 3 hydrogen ions are going to, once they flow through the F0 complex, it's going to rotate the stalk about 120 degrees. Every time it does that, it's going to generate 1 ADP to become an ATP. So again, this is an important ratio for you to know. Every time 3 hydrogen ions flow into it, we get 1 ATP, and it's going to turn a gear that helps generate it. Excuse me. So to help keep up, to to help kind of give an overview of oxidative phosphorylation alone first. Each NADH is going to promote the movement of 10 hydrogen ions across the inner membrane. Excuse me. FADH2 is because complex 2 doesn't move 4 hydrogen ions like complex 1, it's going to do 4 less. So each FADH2 is going to drive 6 hydrogen ions across the inner membrane. And one last ratio to note is that the generation of 1 ATP is going to occur every time 3 hydrogen ions flow through the ATP pump at F0. So for oxidative phosphorylation, it will come back. So in oxidative phosphorylation specifically, for every molecule of glucose, when we have 10 NADH reducing equivalents that are given in, there's going to be approximately 30 ATP generated. Again, because for every NADH, we get 10 hydrogen ions, that leads to, excuse me, about 100 hydrogen ions there. And again, this is all, we're going to talk about why it's approximately 30 in just a moment. We get 2 FADH2s from every glucose, and that'll elicit and lead to about 4 ATP generated as a result of those reducing equivalents. The reason why this says approximately 30 is because as electrons are passed throughout the inner membrane from complex 1 to coenzyme Q to complex 3 to complex 4, electron leak can occur. So as we lose some of these electrons, hydrogen ions will not flow as will not be flowing as much as a result of this electron leak. So because of that, there's never going to be, I shouldn't say never going to be. There is oftentimes not the what truly happens will not oftentimes correspond to what the max theoretical yield is of how many ATP we can get from one glucose molecule during oxidative phosphorylation. So that's why a lot of times you're going to see approximations of how many ATP we can generate from one molecule of glucose. So to bring all of these metabolic processes together, we're going to get 2 ATP from glycolysis, okay? In addition to that, we're going to get 2 pyruvate molecules as a result of that. Excuse me. During the intermediate step, we go from 2-pyruvate to 2-acetyl-CoA. That's going to feed into the TCA cycle. From the TCA cycle, we're going to get 2 more ATP. But the key thing there is that it's going to help us generate reducing equivalents. They're going to feed into oxidative phosphorylation. And from there, the NADHs will help us, the 10 NADHs that we get will help yield approximately 30 ATP from there. The 2 FADH2 reducing equivalents will help generate 4 ATP, producing a max theoretical yield of 38 ATP. Again, this is a max theoretical yield. There's oftentimes electron leak that occurs at mitochondria. Particularly, there's more and more areas of research that are studying mitochondria dysfunction and how that relates to disease. As more and more information is coming about mitochondria dysfunction, we're also understanding more and more about how the disruption of these processes can contribute to disease. There's also whole areas of disease that look at how metabolism affects things like scar formation, how metabolism of heart cells might contribute to either myocardial infarction, heart failure, how metabolic processes can be targets for things like cancer. And so the function of mitochondria is becoming more and more critical in research. But the key thing to note here is that the inefficiency of the passing of the electrons leads to electron leak and can oftentimes does not enable the max theoretical yield typically. I'm going to stop there for a few questions because we just covered a lot and went over everything. So I want to give space for any clarifications. All right. Well, if there's no questions, we can press on. Again, feel free to raise your hand. So the transport of different molecules across these membranes within the mitochondria is really important. The inner membrane is oftentimes impermeable to larger or polar molecules, and so they rely on and depend on the presence of transporters to help shuttle them into the matrix of the mitochondria, where the TCA cycle is occurring. So some of the molecules that we've touched on or talked about, from pyruvate to malate to fumarate to succinate, citrate, or isocitrate, all of these rely on these key transporters at the membrane to help shuttle them into the matrix. So one thing that's really key is, again, glucose is not the only source of energy that can plug into these processes, but amino acids that are either consumed through food, or in cases where glucose is not readily available, oftentimes in instances of either fasting or starvation, muscle wasting can occur, where muscle breakdown occurs, and the protein from your muscles is broken down into its constitutive amino acids, and the amino acids are used to plug into the TCA cycle. For an example, we have alanine that goes under a transamination event to become pyruvate. We have aspartate that undergoes a transamination. So transamination is just the loss of this amine right here. Aspartate via transamination can become oxaloacetate, which is one of the key molecules that is at the top or at 12 o'clock of the TCA cycle, or can go back up the glycolysis pathway for gluconeogenesis. And finally, glutamate via transamination can become alpha ketoglutarate, which is one of the intermediate metabolites in the TCA cycle. So but what's also key is that these processes are reversible. We can go from pyruvate to alanine, we can go to oxaloacetate to aspartate, and we can go from alpha ketoglutarate to glutamate. So this actually enables either the catabolism or the breakdown of these amino acids to feed into the cycle, or the anabolism, the building up of these amino acids from our intermediate metabolites from the TCA cycle. So that is how protein or amino acids can be used as metabolites during these pathways. Another key thing is that lipids can also be used to plug into these pathways via its oxidation. So what ends up happening is, typically in our blood, our lipids are present as triglycerides. It'll break down. Excuse me. These lipids will be broken down via enzymes that are called lipases. Again, anytime you see the suffix ase, that's an enzyme. Lipases, they break down lipids. So lipases will break down the triglycerides into constituent pieces that'll then be metabolized to become acyl-CoA. And so via dehydrogenation event here, where we can generate FADH2, it'll undergo excuse me. It'll undergo a few metabolic intermediate processes that'll eventually lead to the oxidation of this beta carbon at 3-keto-acyl-CoA. So this beta oxidation event is really key, because from here we go from 3-keto-acyl-CoA back to acyl-CoA and acetyl-CoA. The emergence of acetyl-CoA will allow us to plug into the TCA cycle, and it helps generate another acyl-CoA species that we can either be used to generate more lipids, or plug back, or be metabolized again via beta oxidation to generate another acetyl-CoA. So the take-home message is that lipids are that in the form of triglycerides in our blood can be used via beta oxidation to generate metabolites that plug into the TCA cycle. That generates an acetyl-CoA here. And that'll also be generating, additionally, reducing equivalents through this process. One what this looks like all together as kind of a balance between amino acids, lipids, and glucose, is we have our glucose that will move down glycolysis. We have our amino acids that can plug into the TCA cycle via different amino acids being processed differently. We also have, again, our triglycerides that via lipases can break it down, break down triglycerides into a glycerol, into fatty acids. These fatty acids are processed to become, via beta oxidation, from everything from the last slide. That's how we go from fatty acids to acetyl-CoA. So acetyl-CoA can either plug into the TCA cycle, or it can plug into to becoming ketone bodies. That's a structure that we're going to talk about a little bit more in the next lecture. But what's interesting is that also the glycerol here can either be used to generate glyceraldehyde 3-phosphate, which can either plug into glycolysis to produce pyruvate, or feed into gluconeogenesis to generate glucose. I want to stop here and give space for any questions, either on the breakdown of amino acids and fats for metabolism, or again, give space for the overall overview we just had several slides ago. Let's see. No questions. I'm surprised. Thank you.
So, we're talking about how amino acids get up.
Mm-hmm. So what's going to end up happening is in cases where, in diabetic patients where insulin's either not being generated or there's a resistance to insulin, what's going to end up happening is you're going to have the formation of, and I'm glad you asked this because there's one thing I didn't touch on. We can actually go from glucose to glycerol to triglycerides. So once we get to triglycerides here, we can go from fatty acids to these ketone bodies, which is how diabetic patients get into diabetic ketoacidosis. And so that is typically, and I hope I'm answering your question here, that's because of the reversibility of this process here, not just going from triglycerides to glucose, but going from glucose to triglycerides, that enables ingesting glucose in a diabetic patient to generate these ketone bodies. Does that answer your question? And we'll talk a little bit more about ketoacidosis as a general state, but also we can talk a little bit more about how it relates, again, we can explore a little bit more how that relates to diabetic patients in general. Did you have your hand up over there? That hurts. That hurts. Are there any more questions here? Okay, so what I'm realizing now is that I was hoping each year I hope to get to cell cycle and receptor tyrosine kinases. I typically have to end up splitting this lecture in half because of how lengthy it is, and so we're gonna actually forego receptor tyrosine kinases, that lecture, and just wrap up next class with finishing OXPHOS and kind of exploring this a little bit further and how different diets and different states can drive us down these different pathways. I'm going to end again the class with just two quick announcements. So that also means that you don't have to do the pre-lecture for cell cycle and receptor tyrosine kinases. That's not one of the announcements. Again, your final research statement will be due on Tuesday. All of you will, most of you should have your feedback already. That was released this morning. For those of you who are not seeing your feedback yet, that should be finalized by tonight. The last announcement is I am going to my office to go set up that quiz now. I will be going to my office to set up that quiz after this. You'll have until midnight tonight and it'll be set with a 15-minute timer. The questions will not be the same, given that the shoddy version that I made release the answers after you submitted. But again, if you're happy with how you did on the first quiz, you don't have to take it. If you take this one again, please obey the honor code. It's 15 minutes long, and I'll be back. I'll be here if you have any questions. Oh, yes. Yes, great question. Thank you. So I will be maintaining my Monday afternoon from 3 to 4 office hours and my Wednesday from 9 to 11 office hours. Out of respect for the teaching team members who are also students that have their own final exams and commitments, I'm going to let them determine whether they're available. I know, Ahava, you're taking the MCATs next week. 2 weeks, sorry. She will be less available. And so if her office hours work really well for you, I would say reach out to me and I can try and coordinate to find another office hours time, as well as like now my Tuesdays and Thursdays from 2 to 3:15 are going to be open as well, which I don't know if that's going to be an exam time for any of you, but during final exam week, if that's a time that works for you where you want to meet and chat, totally fine. If 9 to 11 seems less appealing. Are there any other questions regarding today's quiz, final research statement, or office hours, or anything else? All right, you're dismissed. I'll if you have any questions about the quiz today, let me know, and I'll see you all on Tuesday.